So welcome everyone to the Sudden Arrhythmia Death Syndromes Foundation webinar on living with a gene-elusive condition. Today is July 22nd, 2025. I'm Marcia Baker, the Program Director of the SADS Foundation. It's my pleasure and honor to introduce our guest presenter for this webinar, Dr. Greg Webster. Dr. Webster is currently Section Chief of Electrophysiology at the Ann and Robert H. Lurie Children's Hospital of Chicago. And he's Assistant Professor of Pediatrics and Cardiology at Northwestern University Feinberg School of Medicine. His educational background includes receiving his medical degree from Stanford University. He received his Master of Public Health at Northwestern University, and this year completed a Master of Healthcare Delivery from Dartmouth College, his alma mater for his bachelor's degree. In Dr. Webster's research lab, he and his team investigate familial causes of heart rhythm problems. His clinical focus is on patients and families with inherited cardiac conditions. Dr. Webster is active with the SADS Foundation as a member of our Scientific Advisory Council. And our staff also had the privilege of partnering with Dr. Webster and his team last November when he hosted our SADS International CME and Family Conferences at Lurie Children's Hospital of Chicago. Following Dr. Webster's presentation, you'll have the chance to type your questions in the chat, and we'll address as many as possible. As a reminder, this webinar is being recorded, so please keep your audio muted. Our team will email you the recording link in a few days. So welcome, Dr. Webster. We look forward to hearing more about a gene-elusive SADS condition.
Thank you, Marcia. Thank you to everybody at the SADS team. You are right. We had a whole team that was helping put together the SADS conference. I would be remiss if I didn't thank and acknowledge my partners, Kendra Ward, Sammy Chowkey, Stephanie Chandler, Ledja Desai, Lindsay Gackenheimer-Smith, Jeff Hong, Sarah Cherney, and Sarah Jurgensmeyer, who just did amazing work in putting it together and are part of how we have such a vibrant center here. I'm going to talk about gene-elusive SADS conditions. And my sense is that I think you guys will feel the same way by the time we're done, that this is surprisingly common in our community. And so I've tried to keep us to 30 or 40 minutes of didactic talk with the hope that we can have a bit of a conversation at the end. Certainly, I know from having met some of you at our patient-focused centers that there are folks on the call that are living with this in a way that I'd love to hear your insight and love to hear the ways that you all think about it. So I'm really excited to join, and I'll kind of jump right in. I do have a consulting relationship with Solid Biosciences, which is a gene therapy company, and I'm going to discuss gene therapy at this during this conversation, but nothing about Solid Biosciences and deliberately nothing about the work that they're doing. So, interesting. Not sure what the scribbling is there. Hang on here. Let me just see if that's... So this is the roadmap that we're going to talk about. I'm going to talk about, first of all, what is monogenetic disease so that we have a way to define what gene-elusive disease is. And then talk about several ways to be gene-elusive. What does it mean to kind of have a gene-elusive disease? And what are the various ways that we talk about it when we do talk about it? And then I'm going to talk briefly about therapy and research, both how are we currently treating genetic diseases that are gene-elusive, and what do we think are the possibilities moving forward? I think everybody on this call probably knows this, but before we get started, I think it's worth saying out loud, what do we mean when we talk about SADS conditions? So this is directly from the SADS website. Sudden arrhythmia death syndromes are genetic heart conditions that cause sudden death in the young. Apparently healthy people, meaning young, apparently healthy people. And these conditions can be treated and deaths can be prevented. So I'm going to talk a little bit about conditions that are kind of iconic for SADS, like long QT syndrome. I'm going to talk a little bit about conditions that do not always lead to sudden arrhythmia death, but can. Arrhythmogenic cardiomyopathy and other cardiomyopathies are part of that. And then I'm going to talk a little bit about diseases that we're not quite sure how to classify them yet. Okay, so this is what people sometimes call the central dogma of biology, which is the idea that DNA gets transmitted to RNA and then into protein. And protein is not kind of protein the way we think of it as a steak or a piece of fish, but this idea that a column of amino acids get folded in a very precise way, a very precise folding mechanism so that they can do a job in the cell. And when we talk about genetic changes, what we mean is that somehow in the process of that DNA duplication or that DNA inheritance from your mother or father to you, that there's a variant and it causes an abnormal protein. And that abnormal protein, instead of getting folded into a perfect paper airplane, gets folded or gets truncated or otherwise gets moved into an abnormal looking protein. The consequences of that are abnormal cell function. And sometimes we're very sure how that happens, and sometimes we're less sure. And that's the process of going from very clear pathogenic genes to gene-elusive conditions. So each parent gives you one copy of each chromosome. So when we talk about diseases in cardiology, we're mainly talking about chromosomes where you get one copy from each parent. And here's a disease called catecholaminergic polymorphic ventricular tachycardia. And it is one of the really well-described SADS conditions where young people especially have disorganized, highly dangerous heart rhythms that can cause sudden death. And in particular, this patient that this tracing is from was a 13-year-old boy who passed out in the middle of exercise. And when we did genetic testing on him, we found a pathogenic variant in the ryanodine receptor. Now this, we're not going to talk about the details of the science, but this is a really clear link between a disease that we know very well, CPVT, a gene that we know very well, the ryanodine receptor, and a disease and a gene that go together. We know that pathogenic variants in the ryanodine receptor cause CPVT, and a variant that we're very sure is pathogenic. So this is the easiest to interpret. And when some people talk about, oh, I've got a SADS condition, and my doctor did genetic testing, and they told me what the reason was, that is what the pathogenic variant means. And in 2025, this is not only helpful in terms of knowing why you might have a disease, but it's often helpful for choosing therapy as well. And so when we think about these variants that are really, really causative or really pathogenic, they can happen in two ways. Either they can be a new mutation, meaning neither of your parents has any copy of the gene, or they can occur because, let me just start that again. Because your parents give you a copy of the gene. And in that case, that new mutation is being determined from a really big set of information, starting with the human genome, which is over 6 billion base pairs long. Of those 6 billion base pairs, everybody, no matter how healthy they are, no matter how ill they are, even if they have a SADS condition, has 3 to 4 million variants when we compare it against a reference genome or a consensus genome that we assemble from what we think of as healthier portions of the population. And then from those millions of variants, there are about 20,000 variants that occur in expressed genes, genes that follow that central dogma of DNA to RNA to protein to cells. And then the numbers start getting smaller, about 300 that cause loss of function, about 15 of those are which are expressed in the heart, and you're narrowing this down to a potential disease-causing variant. And usually you get a report that only lists the variants out of a small panel of genes. So this pathogenic variant that we saw in the ryanodine receptor, in this case, has been culled from an enormous amount of information. And this is the case of the most clear interpretation, but we're going to see as we move forward that it's not always this clear. Okay, so here we are at this new mutation that's been called from this very large amount of data. But sometimes instead of having a new mutation, you have an inherited mutation, a mutation that was passed on, in this case, from an unaffected mother to an affected child. And the reason why that's very important, and the reason why we are always working harder in gene-elusive disease, is because it has implications when you start adding in siblings. So if there is a new mutation and no one else in your family shares it, then you do not need to worry that other members of your family will also have that variant. But if you have a variant that is inherited from one of your parents, in most of the diseases that cause SADS conditions, each of your siblings has a 50-50 chance of also inheriting that variant. And in this example, we're using pedigrees where the squares represent men, the circles represent women, and the lines across represent a couple that had a child together. And then these are the three male, female children of that couple. So this unaffected mom, because she has a circle that's not filled in, had two children that were affected, both of whom had that ryanodine receptor variant, and one son who was unaffected, who has no change in the ryanodine receptor variant. I'm taking the time to walk us through what it means to have a pathogenic variant so that when we talk about gene-elusive variants, we have an idea of what we're comparing it to. So genotype positive disease, it can be helpful. It requires an effective gene disease pairing. In other words, the disease that you're searching for should be one that is caused by the genetic change in question. If you have a pathogenic variant in a gene that has nothing to do with the disease, that's not helpful in interpreting the family. It's useful because it gives a sense of cause and effect for families and for providers. It can be awfully emotionally reassuring to know what the cause is for the disease in a family. It can also be very complicated. Sometimes those emotions are very complicated, especially when there's inheritance or when there are other complex family structures. Knowing the gene and the particular variant can assist with therapeutic choices. And then, as I just showed you in the last slide, it can be useful during family screening. And in 2025 and beyond, it also raises the question of gene therapy, meaning, is there a way that we can change either the products of genes or other cellular components to stop a disease at the genetic level? So when I talk about this idea of genotype positive disease, in a perfect scenario, you'd say, well, that's great. That means that almost everybody who has these diseases, we're going to be able to do all of these things and do all this screening. And so this table, I think, is very instructive. So Arthur Wilde is one of the world's leading investigators in this, and he led a team in 2022 in trying to articulate what percentage of each disease is gene elusive. So if you do a great job of determining what the family disease is, and then you test people knowing all the genes we know in 2022, what are the chances of having a gene elusive result at the end? Well, for long QT syndrome, it's about 10%, meaning that 90% of people we really think have long QT syndrome, we find a genetic change. And for catecholaminergic polymorphic ventricular tachycardia, CPVT, the example I gave you, that number is way down at 30%. 10%, 30%, those are small numbers. But as you start to look through the rest of these diseases, there are high numbers and high percentages of gene-elusive patients. So if you have arrhythmogenic cardiomyopathy, which is a disease that can not only cause a sudden arrhythmia death syndrome, but can also cause problems with your heart muscle and structure, 65% of people with arrhythmogenic cardiomyopathy don't have a gene that identifies why they have arrhythmogenic cardiomyopathy. Now, I will tell you that some of these are uncertain estimates. So when I put this table together, I used the high-end estimate for gene-elusive therapy when this particular paper came out. And so these numbers may not be this high in every circumstance in every population. But I think the idea here is very clear that lots and lots of people are gene elusive. So I've walked us through what is monogenetic disease, and I've walked us through how common gene elusive disease is. And in a second, I'm going to walk us through how often to retest people who are gene elusive. But now we're going to talk a little bit about ways to be gene elusive. So here's a 21-year-old college student who died suddenly during ice hockey practice. And this is a common story. So here the pedigree is showing us an unaffected mom, an unaffected dad, and a single son who died suddenly during ice hockey practice. And he inherited one of these chromosomes from his mom and one from his dad. And they do a post-mortem evaluation and they show arrhythmogenic cardiomyopathy. And in arrhythmogenic cardiomyopathy, the wall of the heart, the muscle of the ventricle is abnormal. So that in unaffected people on the left, the heart muscle is a single strong muscle that squeezes and pumps blood. But in arrhythmogenic cardiomyopathy, the muscle has been replaced by scar and fat, and the wall gets thin and it causes arrhythmias. So this is the case where we're in that higher percentage of gene-elusive disease. So in some people, you would have this information. You would say this is a very clear case of arrhythmogenic cardiomyopathy. The histology, which is microscopic evaluation, and the pathology, which means looking at the heart itself, match a disease that we know causes sudden death. But in this case, there was a negative result, and in fact, a negative result on a very large panel of genes. So does that mean that this patient didn't have arrhythmogenic cardiomyopathy? No, this is still a patient with arrhythmogenic cardiomyopathy. But it does mean that they have gene-elusive arrhythmogenic cardiomyopathy. And in 2025, which is when this case comes from, this is a person whose genetic reason for cardiomyopathy is unclear. And of course, what that means is that then when we scroll back and think about how do we screen his siblings, unfortunately, we don't have a way to tell them we're going to be able to genetically rule in or rule out whether you have arrhythmogenic cardiomyopathy. And so we can screen them with phenotype means, meaning echocardiograms and exercise tests and EKGs and other screening tools for arrhythmogenic cardiomyopathy, but we're not going to be able to use a genetic method of screening them. So are there other ways? There are. I walked you through this complicated method of looking at the human genome and looking at variants, and it's possible that there's something that we missed. Most importantly, it's possible that there are things that we learn over time, and that 5 years from now, 7 years from now, 9 years from now, we may be able to identify a reason for his arrhythmogenic cardiomyopathy that we're not familiar with right now. And so that begs the question, when should you retest? And what does retesting even mean? And the question is, so let's say you have had an event, either because you had a personal event like our first patient who had the CPVT, or because you've had a post-mortem event and now your family needs to be screened. The question is, when do you retest and what does that mean? So first of all, the first step that we recommend to everyone is to make sure that you've been seen by someone who is truly expert at genetic cardiomyopathy. And the good news is there are more and more people who are that way every single year. And the SADS Foundation is a great way to help find people who are invested in genetic myopathies and genetic cardiomyopathies and genetic arrhythmia disease if you're not sure who those people are in your area. I know that I had a hard time finding an orthopedist in Chicago, and there's a million of them. The SADS Foundation is a good way to say, hey, here's where I live and I need help finding someone who's expert in this. And so when we think about retesting after you've found someone who's really expert, this is how we do it at our institution roughly. There are no good guidelines for this. There are several guidelines that suggest that you should reevaluate periodically, but there's no standards that are established. And so what do we say to people? For variants on a written commercial report where we can look through the variants and they're a tractable number, usually 1, 2, 3, 4 variants. We look at those every 2 to 3 years, and we update ourselves on what's available in the literature and whether there's any information about that particular variant. And we recategorize them according to the current methods of categorizing variants. The second thing you can do is you can do genomic analysis, that process that I told you of going from billions of base pairs to millions of variants to thousands of exons to a small number of potential variants. You can do that whole process, sometimes just with the person that you're interested in, sometimes with both of their parents, which can give more information. And if there is a very strong phenotype, meaning you clearly have a SADS type disease, or if you have a syndrome where multiple different organs of the body are involved, we often say, let's do that now. We are at a point in genetic testing where that's often useful when very strong phenotypes or syndromes are present. And if you have done that already, we ask the company or we ourselves go back through that data about every 5 years. Truthfully, some people suggest doing it more often, but in our hands, what we find is that that information has its highest yield when we do it every few years. And in this case, we're not sending new blood or asking the lab to run a new system of counting bases and counting DNA. Instead, we're asking them to look in their computer and do that filtering that I showed you from billions of variants down to a few variants, again from scratch, and say, is there something we know now that we didn't know 5 years ago that is now a potentially causative variant? And then how often is it useful to give another sample and do another test from scratch? I think we're gradually getting to the point where that's less and less useful. As we do genomic analysis, we know more and more. For the last about 20 years, about every 5 to 10 years, there's been a new technique that gives us new information. Years ago, it was adding deletion and duplication testing. Then it was something called next generation analysis. Then it was something called whole exome or whole genome sequencing. Now it's something called long read sequencing. And my guess is that for another 10 to 20 years, there will be a new technology every 5 to 10 years. And it's worth thinking about that periodically. In the field, we sometimes consider that a little of a challenge because someone has to pay for that, unlike the informatic reanalysis, where usually there's a way to get that done without a major cost. When you think about redoing the chemistry of sending DNA, extracting DNA, and doing the work to sequence the DNA, that can be more challenging. But this is kind of a guideline. Take a look at variants unwritten report every 2 to 3 years. Look every 5 years or so at whole genome and whole exome sequencing, and every 5 to 10 years, ask if you have the most recent kind of sequencing in case there's a problem that can be solved with new data. Exceptions. Sometimes you have to do it more often. If there are strong family updates, medical updates, or important new data emerge, then that's the right time to pivot and get more information. So I told you about when there are no candidates variants identified, and then I pulled in how often to retest. Now we're going to talk about variants of uncertain significance. And those of you who have attended multiple of these webinars, you've heard people talk about this multiple times. So I'm going to keep this a little bit brief. I got this case from Angie Calwies, who's a genetic counselor at Rush University Medical Center. She does pediatrics, but she also does adult medicine, and I wanted to have a case that had an adult proband instead of just kids. I practice pediatric medicine. So this is a 56-year-old woman with a clinical diagnosis of long QT syndrome. Her QT interval, which is part of long QT syndrome, was definitely abnormal on several EKGs. She had an abnormal T-wave morphology that goes with long QT syndrome. And her medical history included diabetes, high blood pressure, abnormal cholesterol, and thyroid disease, which is far more common in 56-year-olds than it is in the 5 or 8 or 15-year-olds that we tend to see in the pediatric setting. And here's her family tree. So this family tree has many of the same features that I showed you in the last family tree. Notably, this big red dot is the woman that we're talking about. She's 56 years old. She has a long QT syndrome. And then notice that she has an uncle, her dad's brother, who was found dead at age 22, and the details of that are unknown. That's a suggestion here that there is something going on in the family. She has long QT syndrome. There's an uncle who died suddenly of what could have been a SADS condition, but there's no way to know for sure. And so, of course, the question is, what about these two siblings? What about these two children of hers? And what do we do for them? So she had a genetic test which identified a variant of uncertain significance in a gene called KCNE1. And I'm not going to go through every bit of this with you because I think it gets into too many details, except to give enough information for those of you who are expert or very interested to say, is this a variant of uncertain significance? And I would argue that it is. And so now we have a question of, is this variant really causing her disease or not? And the way I would interpret this is that we don't know, that she has gene-elusive disease, that this variant can't be used to define why she has her disease, and it can't be used to screen her children in particular. And importantly, it doesn't answer why her QTC is so long. So one of the steps to care for her is to go into her past medical history and be really careful that neither any of these diseases nor the medicines that are being used to treat those diseases are causing long QT on their own. And then, as is true for many, many cases of genotype elusive long QT and other diseases, now your only choice is to phenotype test the other family members. And here, the young two sons, these two siblings, are probably the ones at greatest risk because they remain young. Her sisters, of which there are two of them, that's what this two inside the circle means, could also choose to undergo phenotype testing. And in a 78-year-old mom, some people would choose to get tested and some people wouldn't, but that's going to be her choice. And this is partially why it's so valuable to have genetic counselors as part of your team, because these people at very different life stages may think very differently about how they want to do phenotype testing and how much they want to know whether or not what their risks are. Finally, I'm going to talk for a second about a third way of being gene elusive that we talk about a little bit less. If we spend a lot of time talking about variants of uncertain significance, we spend less time talking about polygenic risk. And this is a very famous diagram that originally was published in 2009. And it identifies that there are different kinds of genetic variants. There are some variants, like the ones I talked about at the beginning of the talk, that are pathogenic, that are very rare, and they cause very clear disease, so-called Mendelian disease, after a scientist named Gregor Mendel. And these are very, very rare variants that have a very large effect. And then there are some low frequency variants that are not very common, but that they have intermediate effects. They probably do not cause disease as obviously as these big effect variants do. And we often call these risk alleles or modifier variants. They may not be sufficient to cause disease on their own, but they probably modify the risk of disease. And then way down here in these common variants that happen in 1 to 10% of the population, these variants probably have a very, very small effect. But when you start adding them together in combination, sometimes they can add up to a significant effect. And that's what we're going to talk about here for 1 second, because it's part of gene-elusive disease, is why is it possible that some people are more prone to develop these SADS conditions, even when they don't have a pathogenic variant. So these are summed up in what are called polygenic risk scores. I'm only going to take 1 second to explain polygenic risk scores, but the idea is, instead of looking for very rare variants that may cause the disease, you look at very common variants that happen in the whole population. And then you split the population into people who have the disease and people who don't have the disease. And the red dots are common variants that occur more often in people with the disease. And the blue dot is a variant that occurs more often in people without the disease. And the green dot is variants that occur in both groups. And so you say that common blue dot variation tends to cause no disease, and red dot tends to be associated with disease. and that gives you a sense of where risk is being encoded throughout the population. Where if you have more red dots, you are more likely to be someone who ends up with disease. This is data from one of the folks who works in our group, Tanner Monroe, and his direct supervisors are Meg Puckelwartz and Beth McNally. And this is a figure that was presented at the AHA scientific sessions where we looked at polygenic risk scores in the heart and whether they were associated with sudden death. And so these are polygenic risk scores that were not developed to look at sudden death. And the ones that fall into the colored bands are sudden death risk score, sorry, are risk scores that were not developed for sudden death that are associated statistically with sudden death, suggesting that there is polygenic risk in the genome that is likely to be associated with sudden death that we are not capturing when we send DNA to companies and ask them to tell us about very rare variants. So I want to talk for 1 second about how I put that all together. There's a concept that Dan Roden advanced decades ago called repolarization reserve, which is this idea that your heart has some degree of reserve and that it doesn't go into very bad rhythms very easily. And the technical way to think about this is in this chart that I showed you earlier. But I tend to think about it more like this cartoon, where there's a fence around us that's protecting us. And as long as you can't see over the fence, you're not in very much risk. As long as you're protected by the fence, you are at low risk. But that if you can see over the fence or you're at risk of getting hit by things that are coming over the fence, then you're at risk for a SADS event. And there are multiple ways to have a SADS event. One is to have a pathogenic or likely pathogenic variant that boosts your risk up so that with just one variant, you're up here above the fence and at risk for a SADS event. But what I suspect, and many other people do as well, is that in genotype elusive cases, that it's not just one big pathogenic variant that is boosting you up into a risk category. Instead, there are multiple smaller contributors: environment, modifier genes, polygenic risk, and that all together, those risks confer the same risk or some portion of the risk, and eventually have some people end up here above the fence that protects us and at risk for a SADS event. So that's another way of saying that polygenic risk is still gene-elusive disease. And so this common DNA variation represents another layer of what it means to be gene-elusive. It's still a little challenging to use that in a clinically meaningful way. One of the problems with polygenic risk scores is that they have not been particularly successful at helping us know how to take action. Who should have a defibrillator? Who shouldn't have a defibrillator? Who should be on medicine? Who shouldn't be on medicine? But as we look ahead and say, how do we categorize risk for patients who have gene-elusive long QT syndrome or gene-elusive cardiomyopathy or gene-elusive Brugada syndrome, this may be part of the puzzle of candidate variants, variants of uncertain significance, modifier variants, and then polygenic risk. So here we are in the roadmap. I spent some time talking about monogenic disease. What are pathogenic and likely pathogenic variants? We talked about how common it was and how often to retest. And then I highlighted 3 ways to be gene-elusive. One is just to have a negative genetic test. We just don't identify anything that has us concerned. The second is this idea of variants of uncertain significance that right now, today in 2025, we don't know how to characterize, but that we think have a chance of being something that influences risk of genetic disease. And then finally, this idea of polygenic risk, which is quantifying risk that is in common variation for all people. That means that not everybody is gene elusive in the same way. And so unfortunately, although it would be great if we could say to gene elusive folks, hey, there's just a wait of one more day or one more year or one more decade or one more period of time, and then we're going to know what gene and what variant caused your disease. Unfortunately, I think there's a meaningful number of people for whom gene elusive is going to be true for a while, because instead of looking for a single box to bump them up to the top of the fence, instead what's going to happen is that we need to find each of the small boxes that is increasing their risk. And each of those small boxes is a different kind of research and a different kind of investigation and is going to take time. So I'm going to pivot to therapy and research, which is the last third of this conversation. And one of the questions that I sometimes get from gene-elusive families or families with gene-elusive disease is we hear all of these opportunities for research in people who have specific genetic diseases. Is that coming for us? Are we going to be part of that? Or are we somehow going to be left at the sidelines, not being considered for these? And I think I can give some reassuring news. And so I'm going to spend some time talking about gene therapy because that's the question that I get asked the most right now. But then I don't want to end until I talk about clinical outcomes research, the research that we do to really figure out how do we improve care for people with disease, whether it's in the cutting edge area of gene therapy or the far more routine methods of helping people live with their disease. So this is the single most simplistic way to think about gene therapy. One, you need to package some kind of gene product into a system that gets into the heart. Right now, we use viral vectors. Something called adenovirus-associated vector is the most common one right now. They will change. I suspect they will change quickly over time. But the idea is somehow you need to get the gene product into the body. And then just as importantly, you need to deliver that vector into the heart. You need to get it to the place where SADS disease affects us. And then you need to get it into the cells of the heart. And in fact, it needs to get into the cell and engage the cellular machinery so that it can start either inhibiting other products or producing products of its own, and eventually lead to production of new protein. And if you remember that very first slide that talked about the central dogma of biology, where you end up with DNA encoding RNA, encoding protein, and then encoding cells, you eventually want all of those cells to live in as normal an environment as possible. So this is the idea of gene therapy. And truthfully, it's an oversimplification and there's no surprise there. But the idea is that we want gene therapy to do these things in SADS conditions. And in arrhythmia syndromes, the focus, or one of the focuses, but probably the most important focuses, has been on the direct cause. And I'm highlighting here work largely from the Ackerman lab, who has thought a lot about something called suppression replacement therapy, where the idea is you're going to suppress all of the gene products from a certain gene. KCNQ1, calmodulin, KCNH2 are the examples on this slide. And then you're going to replace it with an entirely normal copy. And here you're focusing on trying to change the fundamental biology in a gene positive patient. So the reason I've put all these up is to say, well, if you're going to do one of these therapies, you need to know what your gene is. You need to know if it's KCNQ1 or calmodulin 1 or KCNH2. And so if you have gene elusive disease, this may be challenging. Now, this is not the only work. If you're curious to read some of the other genetic ideas that are coming, there's a discussion in the Landstrom article at the bottom. But I think the idea here is this is what is coming immediately for genotype positive patients, especially in arrhythmia syndromes. However, in cardiomyopathy syndromes, which are also SADS conditions, there's a great deal of work going on downstream. because cardiomyopathy, oh, I see a spelling error in there. Cardiomyopathy does not specifically work only at the genetic error to myocardial problem. The genetic error tends to cause myocardial overload. There are well worked out compensatory responses, especially from the brain and the kidneys. Those compensatory responses cause cardiac remodeling, and often the presence of symptoms and increased risk for sudden cardiac death don't occur until well after all of those processes have taken place. So a good example of this is work that's being done right now in a trial called MUSIC Heart Failure with Preserved Ejection Fraction, where they're looking at a particular gene called SERCA2A. And in SERCA2A, what they're looking at is not the primary cause of genetic cardiomyopathy, the way we just looked at in the last slide. Instead, what they're looking at is all of the myocardial responses to genetic cardiomyopathy. So that it doesn't matter quite as much why you have your genetic cardiomyopathy. What matters is the myocardial overload, the compensatory responses, and the cardiac remodeling that follow from the fundamental underlying problem that you've got myocardial overload or myocardial injury. This is a very different paradigm for gene-elusive patients. So if you have gene-elusive disease and you're not trying to treat the very specific gene and very specific myocardial process that's occurring, but rather what you say is, what I want to do is I want to work on the downstream problems that occur when the heart gets pushed off balance. Well, now it's less critical that you know what the first gene is. When you realize that these other problems are happening, you can treat these other problems and you can target these other problems with gene therapy. And in this case, it matters far less what the initial genetic event was that started you down this pathway. And in fact, if I scroll out even further away from just heart failure, I would say that this cascade of genetic causes having direct effects on the heart, and then there being compensatory and extracardiac effects because there are problems with your heart. And those compensatory and extracardiac effects then have other problems with your heart. This paradigm opens up a huge world of gene therapy. How do we treat not only the direct causes on the heart, but also the ways that your body adapts to being with a genetic condition? And this is one of the reasons that I remain equally excited for gene therapy for people who have gene-elusive disease as I am for people who have genetic positive disease. And I think of that as a really positive thing. The final thing I'm going to do before I leave us 10 minutes for questions is to point out that gene therapy is the thing that so many people want to talk about right now. But most of the research history in sudden arrhythmia death syndrome has included gene-elusive patients. And I suspect that there are tons of more research projects that are going to happen that have nothing to do with gene therapy and for which gene-elusive patients are not just should be involved, but must be involved. So what drugs are optimal for patients with SADS syndromes? What are optimal ways to monitor for disease progression? How do we select patients for defibrillators? How can we make genomic autopsy universal in SADS conditions? How do we optimize family screening? These are not solved problems in our field. And there are many more conditions like it, and there are many more questions like it, and that's good because I make a living trying to understand these kinds of questions. But I also think it's really, to me, inspiring to know that we don't know everything there is to know about these conditions yet. Remember all the way back to the 10 to 85% of SADS conditions that are gene-elusive. Gene-elusive patients are going to be the heart and soul of SADS research for a very long time. And I think we're going to make very big gains in helping families that don't also have a gene. So here's my summary. It's true. Gene-positive disease is the easiest to interpret. And when you go to meetings and when you go to family gatherings, it is really easy to talk about genotype positive disease because it is the easiest to interpret. But somewhere between 10 and 85% of SADS diseases are gene elusive. You should see an expert in genetic disease before you decide you have gene-elusive disease, and you should make sure that your phenotype has been really well worked out. Phenotype meaning the medical tests that prove what your disease is like. But when you're all done, even with an expert, 10 to 85% of the time, you're not going to have a genetic answer. And we know that. In the hands of an excellent genetic counselor, genetic reevaluation should occur every 2 to 5 years. And then in gene-elusive disease, it is likely that multiple risks combine to determine genetic clinical risk. That's that cartoon of the big pathogenic, likely pathogenic box versus the 3 smaller boxes for multiple risks in gene-elusive disease. And then I am very convinced that there are exciting opportunities for research, new therapies for patients with genotype positive and genotype elusive disease coming. So I want to thank everybody for inviting me. I want to thank SADS. I want to thank all the supporters of the SADS mission and vision. I want to thank families who contributed to the cases that were presented today, in particular, Angie Collies, again, for letting me use her family for the 56-year-old woman. And then my partners and colleagues who worked so hard to make this a center that I'm proud to call home. And then I will stop here and answer any questions that I can.
Thank you very much, Dr. Webster. And we have a few questions that I have received, and then we have one Valerie has put here in the chat. So any other questions, feel free to post them in the chat to everyone. So Valerie asks, are connective tissue disorders such as Marfan's, EDS, what is happening to the research in light of the Doge cuts?
I'll answer the Marfan's and EDS question. The good news and kind of sometimes challenging news is that new genetic conditions we continue to get new insights in genetic conditions. And traditionally, Marfan's disease and Ehlers-Danlos syndrome, which is what EDS stands for, and even coronary artery disease has been a bit outside of what we call SADS conditions. But that's a slippery boundary. If you are a 70-year-old and you die suddenly of coronary artery disease, or you are a 25-year-old who dies suddenly of sudden cardiac death because of genetic coronary artery disease, your family certainly thinks that you had a terrible event. And so if we can help folks like that with genetic data, we want to. Certainly, we would never ask anyone with Marfan's or EDS to step outside of our community. But oftentimes, there are other things that cause Marfan's and EDS to present to clinical care before a sudden death event, and that's good. Just like SADS, there are support groups for both Marfan's and EDS. And although I would say that we would invite anyone into our community, there are also very good support organizations for folks with those diseases as well. What's happening with research in light of the Doge cuts? I will not wade into the politics of this here, meaning like what's a Doge cut, what's a cut from the legislature, what's a cut from the executive branch, but not through the Doge program. What I would say is that the NIH is definitely reconfiguring its priorities right now. I don't know how that will end up. At Northwestern University, which is where I work, there have been substantial cuts, and that has led to some reprioritization of our research work. I will say that as someone who studies SADS conditions, I can probably speak best for myself. And none of the grants that I have that study SADS conditions have been stopped. We are going full speed ahead on the grants that we have. I'm not in a good position to judge where the NIH is going over the long term, but I am so confident that there are both foundations and individuals and national organizations and truthfully the federal government that care very much about these diseases, and that makes me very optimistic.
Great, thank you very much. A question for you. My genetic testing showed I have a VUS. Will my genetic counselor or doctor contact me if there are any updates to my genetic testing results?
So number one, I congratulate you on knowing your own results well enough to be able to articulate that. One of the things we talk about a lot with advocacy organizations is know your own disease. Know what your disease is called, know what doctors think about it as, and then know your own genetic test results. And as it's clear from you, a genetic counselor or someone else has told you about your results already. So the answer is it depends. There are some organizations and some companies that are trying to be proactive about letting people know if the classification changes. The most recent consensus statement from the American College of Medical Genetics puts the responsibility for doing that on your doctor. That is the person who, according to the ACMG, is supposed to update you if that changes. But in the real world, that's something that is still very much different from center to center, and even quite honestly from doctor to doctor. As you saw from our slide, and I will tell you that at least earlier in the call, Sarah Cherney, who's one of our genetic counselors, was on the call. We have infrastructure built by Sarah Cherney, thought up by Sarah Cherney, and put into action by Sarah Cherney to make sure that families get called and get reevaluated periodically, usually every 2 to 3 years. If you don't go to a place that has that system in place, then I would invite you to talk to your doctor every 2 to 3 years, kind of bring it up and say, hey, remember that VUS? What do we need to do in order to update this and be sure that it's still a VUS?
Okay, thank you very much. And Sarah still is on the call. Okay, another question. Should a de novo mutation be tested generations ahead of the parents of an affected child, such as the grandparents?
Yeah, thank you for asking that question. I'll have to make some assumptions to answer that question. I think, and feel free to jump on the chat or email Marcia if I've got it wrong. But I think what you're asking me is if the person who is affected has a de novo variant, should you check that person's parents and should you check that person's grandparents? I'll answer that in 3 separate groups. The first group is usually a de novo variant does not need to be checked in your parents. The slightly more complicated answer is there is some very specific circumstances in which your blood cells that you check can have a variant, and the blood cells in your parents that are checked cannot have a variant, and yet you can still have inherited from your parents. And that's when one part of your parent's body carries the variant and another part doesn't. And in those very specific circumstances, sometimes we will do more checking, and that gets quite complicated, and you would want an expert on your side. But if it's determined that your parents definitely don't have it, then your grandparents do not need to be screened. And that's the third part of it, is even in the super complex circumstance where you may have a parent that has different variants in different parts of their body, nonetheless, you don't need to screen another generation up.
Okay, thank you very much, Dr. Webster. Let's see, any more questions from the group? Here's one that came in earlier from Amanda. Genetic results were inconclusive, and she worries for her 17-year-old son with long QT syndrome. What should she do?
Amanda, if you're on the call and you feel comfortable sharing more about what you're worried about, I'd love to respond most directly. I'll start with a very general response, but if you're willing to talk it through, I'd be happy to answer you directly. This circumstance of having long QT syndrome and being in that small 10% of long QT syndrome that doesn't have a genetic cause, it's frustrating. It's frustrating to be part of that group where genes usually give an answer and they don't. And we certainly have patients that are in that situation at our center as well. The good news is that long QT syndrome is one of our best studied diseases. And if your son has seen an expert in long QT syndrome who has decided that he definitely has long QT syndrome, and has come up with a treatment regimen that makes sense for him. What we know is those treatments in long QT syndrome are very effective for genotype elusive patients as well. And so I would feel confident that if you've built a really reliable treatment plan, even in the setting of being gene elusive for long QT syndrome, my hope and expectation would be that you'd have extremely good results.
Unfortunately, Amanda's not on the call, but it came in through her registration. She asked that question. So thank you very much.
If she watches it on the recording, hopefully that's
she
useful.
will indeed. That's right. All right. Any more questions from the group? Okay, seeing none, I will thank you, Dr. Webster, for your a comprehensive overview here. And I thank all of you in our community for joining us today and asking some very wonderful questions. Let's see what we had several thank yous in the chat for Dr. Webster. So we appreciate your volunteer time and expertise.
I
So thank
appreciate
you.
being invited and thank you all for attending.
All right. Thank you. Have a good evening, everyone.